screen.
And you can share your screen, yeah.
Cool. Sorry if it takes, let's see if, just a sec. There we go. Can you all see that?
There's something big. Yes, I can see it. Yeah, that's good.
Perfect. Thank you so much. All right, with that, then I guess I'll begin. So, hello, my name is Aidan Desjardins, and today I'm going to be talking about non-self recognition in viral infections and its implications in EBV-linked autoimmunity. So first, a little bit about me. I'm a third-year microbiology student at Trinity College Dublin. However, my focus is actually primarily in global health and tropical medicine. And in that respect, I work with the Irish Global Health Network and the Royal Society of Tropical Medicine and Hygiene. And outside of that, I actually enjoy debating and travel with my family. I did have to include a little picture of my dog there. So today's topics. First, I'm going to pose the question, what is the connection between non-self recognition, EBV, and autoimmunity? I'm then going to touch briefly on the methods I use for this literature review. Then I'll contextualize what our knowledge is of non-self recognition, both in the innate and the adaptive immune system. I'll get into what both of those mean in a second. Finally, we'll explore the links between EBV and MS, specifically multiple sclerosis. We'll contextualize the two diseases and then discuss what the current hypotheses are for their connections before moving on to a more broad perspective on the future. So, what is the connection between non-self recognition, EBV, and autoimmunity? In truth, this probably isn't a microbiology question, rather it's an immunology question. However, I think in taking the microbiologist's perspective, we can get a new lens on how to view things. Specifically, the microbiologist's perspective, we typically look at disease in an acute sense, where we look at the immediate infection. However, disease is a lot more complicated than that, and the interplay between how the host reacts, how the pathogen reacts, and what develops is a lot larger, longer spanning picture than what we might give it credit for. And I think in taking a broader perspective, we can explore a lot of different dynamics and maybe even a lot of different new ways to approach disease. And as I'll touch on a little bit later, Epstein-Barr virus, EBV, is a perfect example for this. So to touch briefly on the methods, I performed what we consider more of a soft systematic review. So using large-scale databases such as PubMed and Web of Science to explore the current realm of literature around the subject, I set myself a number of parameters, including a cutoff date of 2000, because immunology is such a rapidly advancing field that anything pre-2000 is largely going to be irrelevant. Further, I focused my search using specific terms, including EBV and MS, as well as specific immunological terms like toll-like receptors, CD8 positive T cell receptors, etc. And then once I created a baseline of literature, I connected this collection of knowledge both through citations and through key terms in order to create a more well-founded and larger body of work. So let's take a look at non-self recognition strategies. And so what I really mean here is, how does the body recognize something other than itself, namely pathogens? And so this comes in two main branches. First of all, you have the innate immunity, that first line of defense, typically non-specific and is found throughout the body. And so what we see here is, in the context of viral infections, you have two key pathways. You have pattern recognition receptors, which recognize specific types or patterns in molecules that you just don't see in humans. And then you also have specific antigen recognition. And what this means is, an antigen is some sort of molecule that your immune system reacts to, typically something that is non-self. And the major pathway in recognizing specific antigens in this context is the major histocompatibility complexes. And what these protein complexes do is they actually present these antigens to the adaptive immune system. And the adaptive immune system is the arm of the immune system that develops and reacts against specific pathogens, specific diseases. In the context of viruses, we have three major arms of this. We have the CD8 positive T cells, which are otherwise known as the cytotoxic T cells. And what they do, as their name may imply, is that they'll detect infected cells and then they'll kill them. They're helped along by B cells, which you've probably heard of in the context of COVID, which produce antibodies. And of course, antibodies are these proteins that can bind to viruses. So what B cells, and actually as an interesting aside for B cells, the actual method of antigen recognition is not really known. We have some ideas that it might involve cross-reaction, but overall the mechanism is just unclear. And of course, with antibodies, as I'm sure you've heard about plenty of times now in the news, what antibodies do is they block viruses from entering cells by binding to those specific those entry proteins, and then also help other immune cells mop up these viruses so they can be eliminated, and what we call neutralizing antibodies. So now let's take a look at EBV and autoimmunity. First of all, Epstein-Barr virus. Epstein-Barr virus is, or EBV, is a double-stranded virus in the family Herpesviridae, and it is extremely common. Over 90% of people on the planet have EBV or contracted sometime in their lifetimes, and for the vast, vast, vast majority of people, they will never express any symptoms. For some people who develop the disease later in life, they might develop what is called infectious mononucleosis, or mono. However, again, that disease usually goes away after a short period of time. However, EBV infection is permanent, like other herpes viruses. And what it does is it actually colonizes our B cells and survives in those B cells. And what can happen is later in life, these viruses might actually reemerge, and which can contribute to more serious diseases such as multiple sclerosis, or MS. And MS is an autoimmune disorder targeting the central nervous system. And essentially what it does is all of our neurons have this insulation, as they're essentially wires, called myelin. And what happens is in MS, the immune system attacks this myelin and breaks it down, leading to a loss of conduction in neurons and eventually cell death. And what we get there is the development of these lesions like you see in this MRI scan to the right. And what should be noted is that this progression is not linear. Rather, it stops and starts as a person ages, which gave us a number of hints in how the disease occurs. So where does this connection come in? In truth, MS is like every other autoimmune disease is a very complicated condition with numerous factors involving it, affecting it, including genes, the environment, and a number of other factors. However, viruses have long been implicated because of their interactions with the immune system. And while EBV has long been thought to be involved, the study that came out just this year, at the beginning of this year, Bjornvik et al., has demonstrated one of the clearest links ever between MS and EBV. This is actually data right to the right from that study. And what it shows is that once a patient contracts EBV, they have a much, much higher risk of developing MS later in life than even if they contract closely related viruses like CMV, a cytomegalovirus. What they found actually is that EBV is associated with a 32-fold increase in risk of MS. No other factor has ever come near that level of association. However, the mechanism is still unclear in this respect. However, there are two big hypotheses on how EBV might actually be implicated in MS. Firstly, you have mistaken self. And both of these reactions involve basically what you can say is a crossed wires in the immune system. And so this first hypothesis basically boils down to, in the development of lesions, both EBV antigens and self-antigens are released as cells die. And what happens there is cytotoxic T cells may instead develop an autoimmune reaction against an immune reaction, an autoimmune reaction against the self-antigen rather than just EBV. It leads to a development of an entire population of T cells that is essentially attacking the body. And one protein in particular, alpha-beta crystallin, has long been implicated as that possible self-antigen. However, I personally find the second hypothesis to be more compelling, and that is called molecular mimicry. And okay, essentially
one more minute now, Aidan, yep. Okay? All right. Yep.
And essentially what this is some sort of host protein has a homology or a similarity or a same sequence as an EBV antigen. And Lance et al., another study that came out this year, found that EBNA1 specifically has a relation to a other CNS protein called glial cam, and they were able to reproduce MS-like symptoms in a mouse model when they were able to induce the production of these specific antibodies. And so what does this mean in the wider scale? As we look towards the future, a lot of work still needs to be done. There is, as I mentioned, there's a lot of factors that are really unclear in non-self recognition at the baseline. However, as we develop more clear mechanisms in the context of EBV and MS, we might actually be able to develop new treatments against MS that target EBV and maybe even possibly a cure. And on the wider scale, I think this is an example that can really show us that disease is a lot more complicated than we give it credit for. And even the most acute illness can have long-term ranging symptoms long into the future. And this is something we should especially be clear of as we gain better perspectives on diseases such as long COVID. So with that in mind, thank you so much for listening. And do you have any questions?
Thank you very much, Aidan. So if you have any questions from the audience, you can just put up your hand or just unmute. Okay, I'll just ask you a quick question. So has this...
You might just stop the recording there, maybe.
Oh yeah, great, yeah, thank you. So many things to remember.